Hi everyone, and welcome to the Chapter 10 video lectures. In this first lecture video, we're going to discuss what DNA is and how forensic analyze DNA evidence. In the previous chapter, you learned about the presumptive and confirmatory tests that forensic biologists use to determine whether or not an unknown substance is a bodily fluid. As I mentioned, the primary duties of forensic biologists revolve around blood examination, either unknown samples from casework or known samples from arrested and convicted individuals. In this lecture video, we'll focus on what modern forensic biology is primarily focused on, DNA typing. Unlike blood analysis done in previous decades, DNA typing allows investigators to individualize the source of the blood to one particular person. This can then be used to link crime scenes, victims, and suspects to one another. Now, DNA is called deoxyribonucleic acid. It's composed of two chains that coil around one another, forming what is called a double helix. The helix chains are formed by alternating sugars and phosphates. Each sugar is attached to a base. The phosphate, sugar, and the base combination are called nucleotides. Each nucleotide has a complementary pair across the DNA helix, with the connections between the bases holding the whole structure together. The bases always connect in matching pairs. Adenine pairs with thymine, and guanine pairs with cytosine. The bases will not connect to one another in any other way, so you won't get adenine connecting to cytosine, for example. It's pretty amazing, but your entire genetic code is spelled out using only these four base pairs. The sequence of bases in your DNA is called your DNA fingerprint. This is essentially a blueprint for your body, telling the cells within our bodies what to do, when to reproduce, how to function, etc. The DNA fingerprint is unique for everyone, and we'll talk more about that in a few slides. The DNA that forensic scientists prefer to use is called nuclear DNA, or nDNA. As the name implies, nuclear DNA is found in the nucleus of the cell. Inside that nucleus are pairs of chromosomes. You can see here how you can unwind the chromosome structure to release the DNA strands. Most people are born with 23 pairs of chromosomes, so 46 total chromosomes. Half of those chromosomes are inherited from your mom, and half are inherited from your dad. All of your chromosomes are contained within the cell nucleus, usually 2 copies of each chromosome within each cell nucleus. Now, you may be wondering how DNA is so helpful for individualization. With only 4 base pairs available, it seems unlikely that this can lead to such a unique genetic profile. Our ability to individualize DNA comes not only from the base pair combination process, but also from the sheer number of base pairs in your genetic code. A single chromosome contains approximately 100 million base pairs. So in your 23 chromosome pairs, you have over 3 billion base pairs. Even with only 4 bases to choose from, this allows for essentially infinite combinations. Another reason that DNA can be individualized so well relates to the mutation potential of our DNA strands. At this time, we can identify the sequence of all the base pairs in your DNA. We call this DNA sequencing, or DNA fingerprinting. The base pairs responsible for making proteins and giving you your characteristics like your eye color, your hair color, etc. This is called coding DNA. For years, scientists believed that the rest of your DNA fingerprint, about 97% of your DNA, wasn't really used by the body for life or development. As we've continued to advance our understanding of DNA, however, the percentage of what we call junk DNA continues to decrease. Since 2003, the Encyclopedia of DNA Elements, or the ENCODE project, has worked to help us understand the functions of our entire DNA system. Some publications from the ENCODE project have claimed to have found a function for up to 80% of the DNA sequence. But many scientists still argue that a large majority of our DNA does not have a coding function that we've found to date. Your textbook makes the estimate of 80% junk DNA, and that's pretty on par with what a lot of people estimate right now. The good news is that even if we don't understand the purpose of all the junk DNA in our bodies, we can take advantage of junk DNA to help individualize DNA to a single person. Because junk DNA is not used to code for something specific, as far as we know, it mutates a lot, and that creates new patterns on a regular basis. In fact, scientists estimate that there are at least 60 new mutations in every living being. Forensic biologists use this high mutation rate to help individualize DNA by focusing on specific areas of the DNA fingerprint that are known to mutate at high rates. Mutation sounds really scary, right? When I say that, like we're envisioning, becoming blue or something. But when we're talking about DNA mutation, we're simply describing a process in which the bases switch places. The advantage of mutation is that mutation creates unique patterns, even in DNA that is directly inherited from your parents. So while you may get half of your DNA from each parent, it's likely that the junk DNA sections will mutate in some way, making your DNA fingerprint unique from your parent's DNA fingerprint. The photograph here shows some of the common forms of DNA mutation. Base pairs can be deleted, they can be duplicated, they can be inverted or spelled backwards, and base pairs can also move between chromosomes entirely while they're being created or duplicated. This process is called translocation. As the slide shows, each mutation can affect a single base, several bases, or a long strand of bases, and these mutations are how your DNA becomes individualizable. To individualize, forensic biologists focus on small parts of DNA strand with high levels of variation. These areas are called short tandem repeats, or STR, and these are typically locations where 4 bases will repeat multiple times. While the sequence itself of the 4 pairs in the STR location does not vary as frequently, the primary highlight for forensic biologists is variation in the number of repeats. The number of repeats in each STR location is affected by the number of repeats from both your mother and your father, so that helps create a more individualized profile. With probabilities, the higher the number of improbable outcomes, the more unique the outcome becomes. So the value of STRs for forensics is not just the probability of the number of 1 repeats in one particular section, but rather the probability of the number of repeats from multiple STR locations all matching up. While each one has more common occurrence individually, when all of the different probabilities are taken together, they provide a much stronger individualization. The FBI developed a DNA database originally using 13 different locations, and when they did that, they noted that the probability of a match of all 13 STR from unrelated, randomly mating populations of Caucasian Americans was 1.74 times 10 to the negative 15th. That means 1 in 575 trillion people. Now, I'll talk more about that system in a minute because that's changed a little bit, but still, huge odds, right? In order to identify the base sequence for each STR, forensic biologists will do what's called an electrophoresis test. Using electricity, they pull apart the DNA pieces based on each piece's size and electrical charge. One of the simplest DNA extraction techniques is through gel electrophoresis. The DNA is put on one end of the gel plate, and electric current is applied to the gel, and this causes the DNA strands to move across the gel plate. Heavier pieces move more slowly, so they won't advance as far down the charged plate as the lighter pieces. This differential movement creates different bands in the gel that can be visualized using a fluorescent dye. These DNA profiles are then compared to unknown samples to determine matches. The picture on the right shows this in a really clear fashion. By taking one sample from the crime scene and separating it into different STR sections, we're able to compare the DNA fingerprint to 3 suspects. The suspect with the matching STR pattern is the person who left the DNA at the crime scene. Now, a much more advanced form of electrophoresis is called capillary electrophoresis, and that works under the same principle of using electricity to separate the DNA, but instead the DNA is separated as it travels inside a small capillary tube filled with a liquid buffer. Capillary electrophoresis has largely replaced traditional gel electrophoresis for a number of reasons, including better resolution and the ability of computer-generated interpretations. This output is called an electropherogram, and it can be used to determine the number of repeats at each STR location. The system that forensic biologists use to store and search DNA samples is called the Combined DNA Index System, or CODIS. CODIS is a national database that contains DNA profiles submitted from federal, state, and local level agencies, and it can be used by investigators to compare against unknown DNA profiles found at other crime scenes. Originally, as I mentioned before, CODIS collected information about 13 unique STRs. These were called the core loci, and you can see these depicted in the chromosome chart on this slide. In 2017, CODIS added 7 more STR locations to the core loci list, providing even higher match probabilities for unknown samples. These new 7 loci are highlighted in blue on the list shown. The additional 7 STR locations are advantageous for the CODIS, because the new locations include segments that can be used with degraded DNA samples, and they also include STR locations that are used by international agencies, so it allows for international comparisons more easily. As of July 2021, the National DNA Index, which is the national level CODIS database, contained over 14.6 million offender profiles, 4.4 million arrestee profiles, and over 1.1 million forensic profiles. That shows you how massive of a DNA database CODIS has become. Most importantly for forensic biologists, though, is the assistance that this database has provided law enforcement in solving crimes and identifying unknown victims. CODIS's primary metric, the investigations aided metric, tracks the number of criminal investigations where CODIS has added value to the investigative process. As of July 2021, CODIS has produced over 570,000 hits that have provided assistance in more than 560,000 police investigations. All right, well, that concludes the first lecture video for Chapter 10. Thanks for watching.